Hello, friend. Welcome to Science for Sleep. Whether you have been here many nights before or are discovering this quiet corner for the first time, this is a place to slow down, learn something gentle, and let the day slowly fade into the background. Tonight, we are traveling back to an Earth without forests. Not simply an Earth where the trees have been removed, but a much older world in which trees had not yet evolved at all. And our way into that world begins with a fossil that scientists have been trying to understand for more than 165 years. Its name is Prototaxites. For much of its history, Prototaxites has been described as a giant fungus. That explanation seems strange enough on its own. Some fossils belonging to this group form tall, thick columns that may have reached about 8 meters above the ground. That is roughly the height of a 2-story house. Yet in January 2026, researchers published new evidence in the journal Science Advances, suggesting that the familiar giant fungus explanation may not be correct. The fossils they examined came from the Rhynie Chert in Scotland, an extraordinary deposit formed roughly 407 million years ago. The Rhynie Chert preserves ancient organisms in remarkable detail, sometimes including structures small enough to be studied under a microscope. Among those fossils is a species called Prototaxites taiti. Researchers compared its internal structure and preserved chemistry with real fungi found in the same ancient rocks. The differences were difficult to explain if Prototaxites was simply an unusual fungus. If you enjoy these quiet journeys, you're welcome to subscribe, and if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. What the researchers found does not mean that scientists suddenly discovered an entirely new kingdom of life. The conclusion is more careful, and in some ways more interesting. Prototaxites taiti had a chemical makeup that differed from the fungi preserved around it. Its internal body also contained arrangements of tiny tubes that did not match the structures known from living or fossil fungi. The researchers suggested that it may instead have belonged to an entirely extinct lineage of eukaryotic life. A eukaryote is simply an organism whose cells contain a nucleus and other complex internal structures. Animals, plants, fungi, and many single-celled organisms are all eukaryotes. If Prototaxites belonged to a separate extinct lineage, then there may be no living organism that gives us a close comparison for what it actually was. That possibility is the latest turn in a very old scientific argument. When Prototaxites fossils were first studied in the 19th century, they were thought to be ancient trees. Later researchers proposed that they might be giant algae. Others argued for fungi. Some suggested something more like a lichen, which is a close partnership between a fungus and a photosynthetic organism. Each idea tried to explain the same strange fossils. They were large. They lacked ordinary wood. They contained networks of microscopic tubes. And they came from a time when the ecosystems on land were very different from anything alive today. The 2026 research has not answered every question. Scientists still cannot point to a living group and confidently say that Prototaxites belongs there. But that uncertainty tells us something important about the world in which it lived. Prototaxites was not standing beneath oak trees, pine trees, or palms. There were no forests filled with trunks and branches. There were no flowering plants. There was no grass spreading across open fields. Many land plants were still small, simple organisms growing close to the surface. The continents themselves were being changed by life in ways that had only recently begun. So the mystery is larger than the identity of one fossil. If an organism, unlike anything alive today, could rise several meters above a landscape without trees, then what exactly surrounded it? What did the ground look like? What kinds of plants and animals were already there? How did rivers behave without deep roots holding their banks together? And how did that strange world eventually become covered by forests? To understand why something like Prototaxites could once seem so enormous and so unfamiliar, we first have to remove something our mind automatically places into almost every landscape on Earth: the tree. The absence of trees covers a much longer stretch of Earth history than it may first seem. The key transition begins roughly 480 million years ago, when the earliest land plants were starting to establish themselves on the continents. It continues through the Silurian and Devonian periods, while plants gradually developed stronger tissues, internal plumbing, deeper rooting systems, and eventually tree-sized bodies. By around 360 million years ago, forests had already become a major part of many terrestrial landscapes. That means more than 100 million years of plant evolution sits inside the phrase "before trees." During that time, the surface of the continents changed repeatedly. Near the beginning, large areas of exposed ground were shaped mostly by flowing water, wind, chemical weathering, and communities of microorganisms. Life already existed on land in forms that could be very small and difficult to preserve. Microbes could live on damp rock and sediment. Fungi or fungus-like organisms were present early as well. So even the oldest landscapes in this story should not be pictured as completely sterile continents waiting for plants to arrive. The first clear land plants added a new kind of biological cover. These early plants were usually short. Many stayed close to moist surfaces because their bodies could not yet move water over great distances. Their growth forms were far closer to small moss-like vegetation than to anything resembling a forest. This stage belongs mainly to the Ordovician period, which lasted from about 485 to 444 million years ago. The Silurian followed, lasting until roughly 419 million years ago. During those tens of millions of years, vegetation became more structurally complex. Some plants developed vascular tissue, which is specialized internal tissue that carries water and dissolved minerals through the body. That innovation allowed stems to rise farther above the ground. By the early Devonian, beginning about 419 million years ago, low vegetation was joined by increasingly upright vascular plants. Some had branching stems. Some produced spores at the ends of those branches. Rooting systems were also becoming more capable of reaching into the sediment beneath them. The landscape containing Prototaxites belonged to this broad phase of change. Around 407 million years ago, when Prototaxites Tahiti lived at Rhynie in Scotland, true forests had still not appeared. Yet the land already supported vascular plants, fungi, small arthropods, freshwater habitats, and organisms with body plans that have no obvious modern equivalent. The world was becoming more crowded and biologically complex long before tree trunks filled the horizon. During the Middle Devonian, beginning around 393 million years ago, another major threshold was crossed. several unrelated plant groups began producing tree-sized forms. This detail matters because a tree is a type of body plan, rather than one single branch of plant ancestry. Tall trunks, crowns, strong support tissues, and extensive water transport evolved more than once. One early tree lineage included the Cladoxylopsids, an extinct group with internal anatomy very different from modern trees. Another later group included Archaeopteris, which developed woody trunks and extensive branching roots. Other tree forms evolved independently in additional plant lineages. There was therefore no single ancestral tree from which every tree-sized plant descended. Evolution repeatedly arrived at the same general solution because growing upward offered access to sunlight and improved the spread of reproductive spores. These changes also mean that different parts of the phrase "before trees" would have looked dramatically different. An Ordovician shoreline with tiny ground-hugging plants would not resemble an early Devonian landscape where vascular stems stood upright among patches of low vegetation. A landscape with towering Prototaxites would have introduced a completely different sense of scale. Later, Middle Devonian environments could contain scattered tree-sized plants, followed eventually by dense stands capable of functioning as true forests. The transition unfolded gradually enough that no single moment cleanly divides a barren earth from a forested one. Each stage added another layer of biological influence to the surface. Plants covered more ground. Their tissues reached higher into the air. Their underground structures became more extensive. By the time forests appeared, the continents had already experienced tens of millions of years of biological preparation. The earliest stage of that transformation is easiest to understand by looking beneath the vegetation entirely. Before deep roots became common, loose sediment, riverbanks, and exposed ground behaved in ways that would make many ancient landscapes feel unfamiliar, even if every living organism were removed from view. Long before forests spread across the continents, the ground itself responded differently to water. During the Cambrian period, which began more than 500 million years ago, and through much of the Ordovician, large river systems often flowed across landscapes with little or no rooted vegetation. Their banks lacked the dense networks of living roots that now hold soil together. There were no fallen trunks wedged across channels. There were no thick mats of leaves building dark forest soil. There was no deep layer of humus, the soft organic material produced as plant remains slowly break down. Much of the surface was exposed mineral sediment. Sand, silt, clay, and broken pieces of rock could lie directly beneath the open air. When rain fell, water struck these surfaces without first passing through a canopy or thick plant cover. Droplets loosened fine particles. Runoff collected them and carried them downhill. Rivers then moved that material through channels that could shift across broad areas of floodplain. The geological record from these early landscapes commonly preserves wide channels filled mainly with sand. Some river systems divided repeatedly around bars of loose sediment, producing patterns that could change after major floods. Wind also had a stronger influence where vegetation was sparse. Large areas of loose sediment could be moved and reshaped because stems and roots were not yet covering much of the ground. Modern landscapes give us a useful comparison. Along a wooded stream today, roots from trees, shrubs, and smaller plants weave through the bank. Those roots act almost like reinforcement inside the sediment. They increase the amount of force needed for flowing water to tear the bank apart. Plant stems slow water near the surface, while fallen branches can trap mud and sand. A treeless bank made mostly of loose sediment has far less of that support. It can erode more quickly when water rises. That does not mean every ancient river was straight, or that vegetation was required for rivers to curve. Meandering rivers, which develop broad bends as they travel across a landscape, can form even where plants are absent. The movement of water and sediment alone is enough to create those bends. Vegetation changes how stable they are. Studies of modern rivers show that unvegetated channels can migrate across their floodplains much faster than comparable channels whose banks are held together by plants. In some cases, the difference can approach an order of magnitude, meaning roughly 10 times faster movement. That gives us a useful way to picture parts of the ancient world. A river bend might slowly shift sideways as water eroded its outer bank and deposited sediment on the inside. Without strong roots, that sideways movement could happen more freely. Floods could rearrange channels. Loose sand could be carried downstream. Mud was less likely to remain locked into thick, stable floodplains for long periods. Even the distinction between river and surrounding land could be less permanent than it is in many vegetated environments today. This physical instability matters because later plants would begin changing it. As vegetation became more widespread during the Silurian and Devonian periods, roots started holding sediment together. Stems trapped particles during floods. Dead plant material began accumulating on the ground. Over millions of years, these biological processes became strong enough to leave recognizable patterns in ancient rocks. That is one of the most important themes in the history of life on land. Organisms eventually became part of the machinery that shapes continents. A root can alter the path of water. A patch of vegetation can change where mud settles. A forest can influence the structure of an entire river valley. But the earliest terrestrial landscapes had very little of that biological control. Their surfaces were still shaped mainly by moving water, wind, exposed minerals, and gravity. Physical erosion could break rocks into smaller pieces and carry those pieces away. It could not create the kind of living ground that later forests would depend on. For that, organisms descended from aquatic ancestors had to begin surviving on exposed land, where water was no longer surrounding them at every moment. The earliest plants to establish themselves on land were tiny compared with the forests that would appear much later. Their beginnings reach back to roughly 480 million years ago, during the Ordovician period. At that time, most complex life still lived in the oceans. Fish had not yet become common in the forms we would recognize later, and no vertebrate animal had begun walking across dry land. The plants making this transition were small enough to remain close to damp surfaces. Their general organization was probably more comparable to modern mosses, liverworts, and hornworts than to any tree. These living groups are known as bryophytes. Bryophytes do not have the complex internal plumbing found in trees and most other modern vascular plants. Water moves through much of their body over very short distances, which helps explain why they usually remain small and are often associated with moist places. The earliest land plants were not exact copies of modern mosses or liverworts. Those modern groups have had hundreds of millions of years of evolution of their own, but they give us a useful sense of scale. A patch of low green growth spreading across damp sediment is much closer to the early terrestrial world than a miniature forest would be. Many of these ancient plants left almost no large fossil body behind. Their tissues were delicate. They did not have thick trunks or heavy woody stems that could easily survive burial. Some of the most important evidence comes instead from microscopic spores. Spores are reproductive cells that can grow into a new organism under suitable conditions. Unlike seeds, they contain no large stored food supply and no developing plant embryo enclosed within a protective seed coat. Early land plant spores developed unusually tough outer walls. Those walls could survive long after the soft plant that produced them had disappeared. Fossil spores found in Ordovician rocks provide some of the earliest evidence that plant-like organisms were already reproducing on land. Damp environments would have offered important advantages. Water could collect along lake margins, river edges, wet sediment, or shallow depressions in the landscape. Remaining close to moisture reduced the distance water had to travel through the plant. It also helped with reproduction, because many early plant groups depended on water for the movement of reproductive cells. These low-growing plants had no tall canopy above them. Their photosynthetic tissue stayed close to the surface where water was easiest to obtain. Photosynthesis is the process that uses light energy to combine carbon dioxide and water into sugars. Those sugars provide chemical energy and building material for the plant. Moving that process onto land opened an enormous new habitat. But the land created problems that aquatic ancestors had rarely faced. Drying was one of the most immediate. In water, exposed cells remain surrounded by moisture. On land, water can evaporate directly into the air. Sunlight also brought greater exposure to ultraviolet radiation. Ultraviolet light carries enough energy to damage important molecules inside cells, including genetic material. Gravity became more important as well. Water supports much of an aquatic organism's weight. A body growing upright on land has to support itself. Nutrients created another challenge. Minerals such as phosphorus and potassium may be present inside rocks and sediment, yet a simple plant cannot easily reach or dissolve all of them. Reproduction had to work in air while still protecting delicate cells from drying. Each of these pressures placed limits on how far the first terrestrial plants could spread and how large they could become. Even so, their importance was much greater than their size suggests. They began adding fresh organic material to exposed land. Their surfaces held moisture in small patches. Their remains mixed with mineral particles after death. Their living tissues also created new places for fungi, microbes, and tiny animals to interact. The continents were beginning to develop a biological skin. That first green cover stayed close to the ground because the basic problems of life on land were still only partly solved. Keeping water inside the body, exchanging gases with the air, protecting reproductive cells, and eventually moving water upward would require a new set of adaptations before plants could rise much higher. Life on land demanded a new way of managing water. For an aquatic ancestor, moisture was always nearby. On exposed ground, every surface facing the air could lose water through evaporation. Early land plants therefore benefited from a thin protective coating called a cuticle. The cuticle is a waxy layer covering the outside of plant tissues. Because wax does not mix easily with water, this coating slows the movement of water from moist cells into dry air. Even a very thin cuticle could make the difference between remaining hydrated and drying too quickly under sunlight or wind. A completely sealed surface would create a different problem. Plants build sugars through photosynthesis, and that process requires carbon dioxide from the atmosphere. Carbon dioxide has to enter the plant somehow. Many land plants solve this with tiny openings called stomata. A single opening is called a stoma. In modern plants, specialized cells around each stoma can change its width, helping control the movement of gases and water vapor. Carbon dioxide enters through these pores and reaches photosynthetic cells. At the same time, water vapor can escape. This creates one of the basic challenges of plant life on land. A plant needs contact with the atmosphere to collect carbon dioxide, while every open pore creates another route through which water can be lost. Modern trees still manage this balance hundreds of millions of years after the first terrestrial plants faced it. Early land plant lineages developed their own versions of these gas exchange systems. Fossils show stomata in very ancient plants, although different early groups did not all use them in exactly the same way. Water protection also mattered during reproduction. Many early plants reproduced using spores rather than seeds. A spore is usually a single reproductive cell surrounded by a protective wall. One especially important material in those walls is sporopollenin. It is extremely resistant to drying and chemical damage. Its toughness is one reason ancient spores can survive in rocks long after the soft plant that produced them has disappeared. Spore walls therefore helped plants send reproductive material through air and across exposed surfaces. Moisture still remained important once those spores began new generations. In many early plant groups, sperm cells needed a thin film of water to swim toward an egg. Modern mosses and ferns still preserve versions of this reproductive dependence. A damp patch of ground could therefore be much more useful than a dry exposed surface, even after plants had developed better protection from evaporation. Remaining small helped as well. A plant only a few centimeters tall can move water over short distances. Some movement can occur directly from cell to cell. Gravity creates little difficulty when the upper tissues are only slightly above the wet ground. Greater height changes the problem. Water becomes heavy when it must be lifted through a long stem. The plant also needs enough internal structure to prevent that stem from bending or collapsing. Wind applies additional force as more of the body rises into moving air. These limits help explain why the first green cover remained low for so long. A waxy surface reduced water loss. Gas exchange pores allowed carbon dioxide to enter. Tough spores protected reproductive cells during part of their journey. Together, these adaptations made terrestrial life increasingly practical, while keeping most early plants physically close to their water supply. Survival above the waterline had become possible. Growth across larger areas still depended on resources beneath the surface. Phosphorus, potassium, calcium, and other useful elements were often locked inside mineral grains. Simple rooting structures could touch those minerals without necessarily extracting enough from them. Early plants gained a powerful advantage when extremely thin fungal filaments began exploring those tiny spaces alongside them. Those filaments belonged to fungi, organisms that were already becoming important partners in the early ecosystems of land. A fungus grows through its surroundings using microscopic threads called hyphae. A single hypha can be only a few thousandths of a millimeter wide. That small size allows it to enter spaces between mineral grains that would be difficult for a larger plant structure to reach. As many hyphae spread outward, they form a branching network called a mycelium. For a small early land plant, that network could greatly increase the amount of ground being explored around it. One of the most valuable materials hidden there was phosphorus. Plants need phosphorus to build cell membranes, store and transfer energy, and make genetic material. Yet much of the phosphorus in fresh rock is chemically bound inside minerals. Simply growing beside a grain of rock does not automatically make that nutrient available. Fungi can help change the chemistry around those grains. Some release organic acids and other compounds that react with minerals. These reactions loosen certain elements from the mineral structure, allowing dissolved nutrients to enter surrounding water. Hyphae can then absorb some of those nutrients and move them through their network. A plant associated with the fungus gains access to a much larger underground feeding area. The fungus receives something valuable in return. Plants use sunlight during photosynthesis to build carbon-rich sugars. Some of those carbon compounds can be transferred to fungal partners growing in or around their tissues. The plant gains mineral nutrients. The fungus gains energy-rich carbon that originally came from the atmosphere. Relationships of this general kind are known as mycorrhizal associations. The word mycorrhiza means fungus root, although some of the earliest plants had simpler underground structures rather than the true roots found in later plants. Fossils show that these partnerships are extremely ancient. Early Devonian plants preserve fungal filaments growing within their tissues, along with structures that appear suited for exchanging nutrients. This evidence comes from a time more than 400 million years ago, long before forests contained familiar trunks and deep root systems. The partnership probably helped early plants establish themselves on land where useful nutrients were scattered through poorly developed soils and weathered rock. Fungi also became part of the slow process that changed the ground itself. Chemical reactions around fungal filaments contributed to the breakdown of minerals. Microbes altered nearby material. Dead organisms added carbon-rich remains after they decomposed. Over long periods, these biological effects mixed with sand, clay, and fragments of weathered rock. Soil was already beginning to form before true trees appeared. Those early soils would have looked different from the deep, dark forest soils familiar today. They contained less plant litter and lacked the dense woody roots that later mixed and stabilized much larger volumes of ground. Their development came from many processes working together, including physical weathering, microbial activity, fungal growth, and the remains of early plants. This gradual change had an important effect on the spread of vegetation. A patch of land that had already been biologically altered could hold nutrients and moisture differently from fresh exposed rock. Plants growing there could contribute more organic material when they died. Fungal networks could continue moving through the developing substrate. The surface became increasingly suitable for more life. Even with these underground partnerships, the vegetation above remained low. Most early plants still lacked a reliable way to carry large amounts of water through a tall body. Their tissues stayed close to the surface because short transport distances were easier to manage. The landscape would only begin gaining real height when plants developed specialized internal tubes capable of moving water upward against gravity. As early plants spread across damp ground, a new kind of tissue began changing how far their bodies could reach above the surface. These plants are called vascular plants. The word vascular refers to an internal transport system that moves water, dissolved minerals, and other materials through the body. One of its most important tissues is xylem. Xylem carries water upward from the lower parts of a plant. Inside early vascular plants, some of the water-conducting cells were called tracheids. A tracheid is a long, narrow cell with strengthened walls. When these cells die, their hollow interiors remain connected as part of a pathway through which water can move. This gave plants something that their smaller ancestors largely lacked: a reliable way to supply tissues located several centimeters above wet ground. Water entering near the base could travel upward through the stem instead of moving only from one nearby cell to another. The walls of these conducting cells also provided extra support. Many contained lignin, a tough substance that stiffens plant cell walls and helps prevent water-carrying tubes from collapsing inward. That combination of transport and reinforcement became increasingly important as plants grew taller. One familiar fossil example is Cooksonia. Cooksonia lived during the Silurian and early Devonian, with fossils dating back more than 420 million years. Its body would look remarkably simple beside a modern plant. Thin, upright stems divided into branches, often splitting into two near equal parts. At the ends were small reproductive structures called sporangia. A sporangium is a structure that produces and releases spores. Cooksonia had no broad leaves forming a crown. Its underground structures were also far simpler than the deep branching root systems that trees would later develop. Many specimens were only a few centimeters tall. Even that modest height represented an important change. Photosynthetic tissue raised above neighboring growth could receive light with less shading. Reproductive structures held higher in the air could release spores into moving currents, allowing them to travel farther from the parent plant. A few extra centimeters could therefore offer real advantages on a landscape where most vegetation remained very low. Height also created new costs. Every additional length of stem placed the upper tissues farther from their water supply. The stem had to resist gravity while remaining upright. Moving air could bend it, placing stress on supporting tissues near the base. As plants became larger, the demand for water increased because a greater surface area was exposed to the atmosphere. The vascular system helped solve these problems well enough for natural selection to keep experimenting with larger bodies. Later, Devonian plants developed thicker stems, more elaborate branching patterns, and increasingly effective conducting tissues. Some began producing true leaves or leaf-like structures with much greater photosynthetic area. Others invested more heavily underground as roots became capable of reaching deeper sediment. These changes unfolded over tens of millions of years. The earliest vascular plants remained small enough that a person could have stepped over many of them without difficulty. Their importance came from the new possibilities hidden inside those slender stems. A plant no longer needed every photosynthetic cell to remain almost directly beside its source of water. Water could be collected below and delivered upward. Minerals gathered from the ground could reach growing tissues farther away. Reproductive structures could be lifted above the surrounding vegetation. The terrestrial surface was beginning to gain a vertical dimension. That extra height also changed the environment for organisms living closer to the ground. Stems created small, sheltered spaces. Dead plant tissue supplied food for decomposers. Spores and other organic material became available to animals capable of feeding on them. By the late Silurian, while vascular plants were still modest in size, some of the earliest known land animals were already moving among this low vegetation. They belong to a group whose descendants still walk across soil, bark, and fallen leaves today: the arthropods. The growing cover of small plants created opportunities for animals that could tolerate life away from open water. Among the earliest successful groups were arthropods, the broad family that today includes insects, spiders, centipedes, millipedes, and crustaceans. Their presence on land reaches at least into the late Silurian, more than 420 million years ago. One especially important fossil comes from the Scottish island of Carrera. It belongs to an extinct animal called Campicaris obanensis. Campicaris lived about 425 million years ago and measured roughly 2.5 centimeters long. Its segmented body resembled that of a small modern millipede, although it belonged to an extinct group rather than the direct ancestry of millipedes living today. Researchers dated the volcanic minerals in rocks surrounding the fossil, giving Campicaris an unusually precise place in the history of terrestrial animals. When the age was reported in 2020, it made Campicaris the oldest known land animal body fossil to have been dated this way. Older tracks and burrows suggest that arthropods may have explored land earlier. Those traces preserve movement rather than the animal itself, so the identity of their makers is harder to determine. By the time Campicaris was moving around Carrera, however, some arthropods were clearly equipped for life on exposed ground. Their body structure provided several useful advantages. An arthropod carries its skeleton on the outside. This hard outer covering is called an exoskeleton. It supports the body against gravity, while also creating a barrier that can reduce the loss of water from softer tissues underneath. That protection became especially useful in air, where an unprotected surface can dry much faster than it would underwater. Myriapods were among the early animals taking advantage of this design. The name refers to the larger group containing millipedes and centipedes, along with several smaller living groups. Early forms often had long segmented bodies with repeated pairs of legs, allowing them to move across uneven plant debris and damp sediment. Food was beginning to accumulate around them as well. Every generation of terrestrial plants left behind dead tissue. Spores settled onto the ground. Microbes and fungi grew through decaying organic material. Animals able to consume this material became detritivores. A detritivore feeds on dead and partly decomposed organic matter, helping break it into smaller pieces. Campicaris has been interpreted as a likely feeder on decaying vegetation around lakeside environments. This kind of feeding created an important link in the first terrestrial food webs. Nutrients locked inside dead plant tissue could pass through animal bodies and return to the surrounding ground. Fungi and microbes continued decomposition at still smaller scales. As arthropods became more diverse, predatory forms could feed on other small animals, adding additional connections to these early communities. The land was therefore developing recognizable ecological relationships long before anything resembling a modern forest canopy existed. Much of this activity probably remained close to the ground. Small bodies could shelter beside stems, within cracks, and among damp patches of decaying material. conditions near the surface were often less drying than those higher in moving air. Vertebrates followed a very different timetable. The ancestors of amphibians, reptiles, birds, and mammals were still living in aquatic environments, while myriapods had already established themselves on land. Fish would continue dominating vertebrate evolution through much of the Devonian. The famous fish like Tiktaalik, for example, lived around 375 million years ago, roughly 50 million years after Campocaris. By then, terrestrial vegetation had changed enormously. The contrast shows how early arthropods arrived during the development of land ecosystems. Tiny animals were already feeding, moving, and reproducing among vegetation that remained low enough to lack anything like a true forest. Their small size also makes their fossil record difficult to read. A body only a few centimeters long can disappear almost completely after death, unless burial conditions are unusually favorable. Soft internal tissues decay especially quickly. Even hard outer coverings are often broken apart or scattered. One early Devonian site escaped much of that loss. At Rhynie in Scotland, mineral-rich hot spring waters preserved plants, fungi, and tiny animals with details sometimes reaching the level of individual cells, leaving one of the clearest windows into life on land, roughly 407 million years ago. The small animals of the early Devonian usually left behind only scattered pieces of their bodies. At Rhynie, in Aberdeenshire, in northeastern Scotland, something very unusual happened to an entire ecosystem. About 407 million years ago, this area contained springs heated by volcanic activity beneath the ground. Hot water moved through buried rock and picked up large amounts of dissolved silica, the same silicon-rich material that forms minerals such as quartz. When this mineral-rich water reached the surface and cooled, silica began coming out of the water. Plants and other organisms growing near the springs could become surrounded by it. In some cases, silica entered spaces inside their tissues before those tissues had time to collapse. Over time, the material hardened into a fine-grained rock called chert. The result is known as the Rhynie chert. Most fossils preserve only hard parts, flattened impressions, or mineral replacements that have lost much of the original anatomy. Rhynie can preserve structures at the scale of individual cells. Thin slices of the rock placed under a microscope can reveal the internal plumbing of a plant stem, fungal threads passing through tissue, or the outer covering of a tiny arthropod. Some plants were even preserved upright in the positions where they had been growing. One of the best-known plants from the site is Rhynie. It stood only a few tens of centimeters tall. Its simple green stems rose from horizontal structures close to the ground and divided as they grew upward. Rhynie had vascular tissue for moving water internally, yet it lacked the deep roots and broad leaves of later plants. Another plant called Asteroxylon had a more complex body. Small leaf-like structures covered parts of its shoots, and its underground system spread through the sediment. These plants grew in an environment that could be disturbed repeatedly by hot spring activity. Mineral-rich water sometimes flooded patches of vegetation, preserving one community, while new growth later established itself nearby. Fungi were woven through this ecosystem at microscopic scales. Some occupied dead plant tissue and helped break it down. Others formed close associations with living plants. Fossil structures inside Rhynie plants provide some of the oldest detailed evidence for relationships similar to the partnerships that modern plants form with nutrient-gathering fungi. The animals were equally small. Mites moved through the vegetation and organic debris. Myriapods were present, along with primitive relatives of insects. Freshwater areas contained small crustaceans and other organisms adapted to pools around the spring system. Their remains give scientists a rare chance to examine how plants, fungi, and animals interacted during an early stage of terrestrial ecosystem development. Even reproductive structures can survive. Spores remain visible inside some fossil plants. Fungal reproductive bodies occur within ancient tissues. Parts of arthropod mouth structures and digestive systems can sometimes be studied in remarkable detail. This level of preservation makes Rhynie especially valuable because most early Devonian fossil sites provide only pieces of the same story. A stem might be preserved without its internal cells. An animal may leave a track without its body. At Rhynie, many parts of the ecosystem occur together in the same rocks. Scientists can therefore compare organisms that actually shared the same environment at roughly the same time. That became especially important in 2026. Researchers examining one unusual Rhynie fossil did not have to compare it only with distant relatives or modern organisms. They could compare its preserved chemistry and anatomy with genuine fungi fossilized beside it more than 400 million years ago. The organism was Prototaxites taiti, the smaller Rhynie relative of those enormous column-shaped fossils that once reached several meters above the ground. Its extraordinary preservation gave scientists a new way to revisit a debate that had begun in the 19th century. And what they found made the old description of Prototaxites as a giant fungus much harder to maintain. The strange history of Prototaxites began long before scientists had microscopes powerful enough to study its finest structures. During the 1850s, large fossilized columns were being examined from rocks in Canada. They looked enough like ancient woody trunks that the paleontologist John William Dawson interpreted them as a primitive tree related in some way to yews. That early idea helped produce the name Prototaxites, a name that roughly carries the sense of a first or primitive yew-like plant. The interpretation did not last. When scientists cut thin slices through the fossils and examined their interiors, they found nothing resembling ordinary tree rings or the organized wood of familiar plants. Instead, the interior was packed with microscopic tubes. Some were extremely narrow, others were wider. They crossed, branched, and sometimes reconnected inside a dense network that looked very different from normal wood. During the following century, Prototaxites moved repeatedly through the biological family tree as scientists tried to explain those tubes. An interpretation as a giant alga gained support for a time. Later studies favored a fungus. A lichen-like organism was also proposed, meaning a partnership in which a fungus lived closely with a photosynthetic partner such as an alga. The fungal explanation eventually became especially influential. It appeared to fit several clues, including the tubular internal structure and chemical evidence suggesting that Prototaxites may have obtained carbon from other organic material, rather than producing all of its own food through photosynthesis. That would make it more fungus-like in the way it fed. The scale remained remarkable. Across the genus, the largest known Prototaxites fossils formed columns reaching roughly 8 meters tall. Some were more than a meter across. They grew during the early Devonian, when much of the surrounding land vegetation remained measured in centimeters. A Prototaxites column could therefore rise several times higher than many of the plants around it. The smaller species preserved at Rhynie gave scientists something the giant specimens usually could not provide. Its tissues were preserved finely enough for researchers to study both microscopic anatomy and traces of its original chemistry. The 2026 team examined those chemical traces using methods that measure how molecules interact with light. Different kinds of biological material leave slightly different patterns. These patterns can work like molecular fingerprints, allowing researchers to compare ancient tissues even after more than 400 million years of alteration. Rhynie provided an unusually strong test because genuine fossil fungi were preserved in the same rocks. The researchers could compare Prototaxites tahiti with organisms that lived in the same environment and experienced similar burial conditions. The match expected from a fungus did not appear. Its preserved chemical composition differed from the fungi surrounding it. Its tube system also failed to match the organization known from living fungi or well-understood fossil fungi. Those two lines of evidence made the fungal explanation much less convincing. The researchers concluded that Prototaxites is currently best placed in an entirely extinct lineage of eukaryotic organisms. Eukaryotes are organisms built from complex cells containing a nucleus, a huge group that includes plants, animals, fungi, and many microscopic forms of life. An extinct eukaryotic lineage would mean that Prototaxites belonged to a branch whose members eventually disappeared completely. No living organism would provide an exact comparison. That conclusion still leaves important details unresolved. Scientists do not yet know exactly where the lineage belongs among other eukaryotes. Its complete life cycle remains uncertain, along with the way its giant forms grew and obtained enough resources to support such large bodies. The 2026 work narrowed the possibilities rather than filling every gap. One feature needs no uncertain classification to appreciate. Long before true forests covered the continents, life had already found a way to build a body several meters above the ground. In a landscape where ordinary plants were still low, that height would have made Prototaxites one of the most physically dominant organisms in sight. Its strange form belonged to a period when the vertical space above the land was still wide open for evolution to explore. The scale of Prototaxites becomes easier to appreciate when it is placed beside the plants growing during the same broad part of the early Devonian. Many vascular plants of that time remained only a few centimeters or a few tens of centimeters tall. Rhynia, preserved at the Rhynie Chert around 407 million years ago, commonly reached heights of roughly 20 centimeters. Its upright stems were simple and leafless, with branching tips that carried spores. Other early plants were beginning to develop more complex shoots and underground structures, but the landscape still lacked anything like a continuous forest canopy. Against that low vegetation, the largest members of Prototaxites would have been extraordinary. Some column-shaped fossils assigned to the genus reached roughly 8 meters in height and more than a meter across. An organism that tall could stand 30 or 40 times higher than many nearby vascular plants. The comparison does not mean that every Prototaxites individual was enormous. Different species and specimens varied greatly in size, and the much smaller Prototaxites tahiti from Rhynie belongs to the same broader group without approaching those giant dimensions. The largest fossils show what the body plan was capable of achieving elsewhere during the Devonian. The land around them would still have been unusually open by modern standards. There were no broad crowns of leaves joined together overhead. Shade would have been limited and patchy. Wind could move across low vegetation with relatively few tall obstacles. Sunlight reached the ground directly across much larger areas than it does beneath a modern woodland. A multi-meter organism rising into that open air occupied a position that trees would later dominate almost everywhere they could grow. Exactly what advantage Prototaxites gained from height remains uncertain. If it released spores or other reproductive cells into the air, elevation could have helped those structures enter stronger air currents. Greater height might also have changed exposure to light, moisture, or moving air. Those possibilities remain hypotheses because scientists still do not know enough about its biology to identify one clear reason for its size. Its feeding strategy is equally difficult to reconstruct. Earlier carbon isotope studies suggested that Prototaxites obtain carbon from organic material rather than relying entirely on ordinary photosynthesis. That result helped strengthen the old fungal interpretation. The 2026 evidence weakened the idea that it was actually a fungus, while leaving the exact source of its food unresolved. Researchers therefore have to separate what the fossil clearly shows from what its missing biology leaves uncertain. There are also signs that Prototaxites interacted with other organisms. Some fossils contain narrow tunnels that may have been made by small arthropods feeding within the tissue. Other specimens preserve internal structures associated with microorganisms. These traces suggest that Prototaxites could itself become habitat or food within early terrestrial ecosystems, although the details probably varied between species and environments. It would also be misleading to imagine endless forests of giant Prototaxites columns. The fossil record does not support a single worldwide landscape covered densely by them. They occurred across a wide geographic range, but the abundance and spacing of individuals differed from place to place. Some Devonian environments may have contained scattered tall columns among much lower vegetation. Others preserve smaller forms or no Prototaxites at all. That uneven distribution makes the world more realistic and more interesting. Evolution was experimenting with several ways of occupying the new space above the land. For a time, one of the most impressive solutions belonged to an organism whose closest relatives may now be completely extinct. Yet height alone would not determine the future of terrestrial ecosystems. Plants were developing an advantage that reached in the opposite direction. Their underground structures were becoming larger, more branched, and better able to enter the sediment beneath them. As those early roots expanded, they began changing the physical ground on which every later forest would depend. The earliest underground structures of land plants were extremely simple. Some plants use thin structures called rhizoids, which helped attach the body to damp sediment and absorb small amounts of water. Other early vascular plants produced horizontal stems called rhizomes that ran along or just beneath the surface. Fine hair-like structures could extend from them into the surrounding material. During the Silurian period, more than 420 million years ago, these shallow systems usually penetrated only a few centimeters into the ground. Over time, plants developed increasingly complex rooting structures. The transition unfolded in several plant groups, rather than appearing once in a single ancestor. Evidence suggests that major changes in rooting systems were underway around 430 million years ago. By the early Devonian, roots were reaching tens of centimeters into some ancient soils. The roughly 407 million-year-old Rhynie chert even preserves some of the oldest known growing tips of true rooting axes. These tips contained a region called a meristem. A meristem is a group of cells that keeps dividing, allowing a root to grow forward through the ground. This gave plants a much more active way of exploring the material beneath them. A branching root system could reach water stored between grains of sediment and contact mineral surfaces, far from the base of the stem. Roots also provided stronger anchorage. A taller shoot creates greater leverage when wind pushes against it. Spreading underground structures help transfer that force into the surrounding ground, allowing the plant to remain upright as its body becomes larger. Roots also began changing the rocks and sediments they entered. Growing tips could follow existing cracks and expand into small openings. Repeated growth helped loosen mineral fragments. Around living roots, chemical changes could be even more important. Roots release carbon compounds into the surrounding ground. Their cells respire, producing carbon dioxide. Fungi and bacteria concentrated around roots add their own chemical activity. This biologically active region around a root is called the rhizosphere. Water containing carbon dioxide becomes mildly acidic. Plants and microbes can also release organic acids. These substances react with minerals and help free elements that organisms need. Phosphorus can be released from phosphorus-bearing minerals. Potassium, calcium, and other elements can also enter soil water as rocks slowly weather. Some of those dissolved nutrients are then absorbed by roots and carried upward through the vascular system. The effect extends beyond feeding the plant. Roots introduce organic carbon below the immediate surface. Tiny root hairs die and are replaced. Larger roots eventually decay. Sugars and other compounds leak or are deliberately released into the surrounding soil, where microbes can use them as food. Ancient soils began recording these changes. A fossil soil is called a paleosol. Silurian paleosols generally preserve shallow plant traces. Early Devonian examples can contain much deeper rooting and stronger signs of biological activity. They also tend to show more intense chemical weathering than many older soils. Clay minerals accumulated as original rock minerals were altered. Organic matter became mixed deeper into the ground. The zone affected by living organisms gradually thickened. This transformation did not happen everywhere at the same speed. Some early and middle Devonian environments still contained poorly developed ground with few recognizable root traces. Other places supported plants whose underground systems were becoming increasingly elaborate. The continents were becoming a patchwork of different stages in plant colonization. As those rooted areas expanded, sediment became harder for moving water to remove. Thousands of roots passing through a riverbank could bind loose particles together. Vegetation growing across a floodplain could survive long enough to trap additional sediment around itself. The influence of plants was spreading beyond the tissues of the plants themselves. By strengthening the ground, roots began altering where rivers could move and how quickly their channels could shift across the landscape. The growing forest world was beginning to leave its signature in the paths of flowing water. As rooted vegetation spread across floodplains, rivers began encountering banks that were harder to move. A river erodes its channel by pushing water and sediment against the material along its edges. loose sand can be carried away relatively easily. Mud is more resistant, and living roots increase that resistance further by tying particles together below the surface. Modern riverbanks show how strong this effect can become. A network of roots crossing through soil works much like reinforcement inside a wall. Individual roots can stretch across weak points, while thousands of smaller roots help keep pieces of sediment connected when flowing water pulls against them. The result is a bank that can survive stronger currents before it begins to collapse. Vegetation also changes what happens during floods. When water spills out of a channel and spreads across a floodplain, stems create drag. The water slows around them. Fine particles that had been suspended in the current can then settle onto the ground. Mud that might otherwise have continued downstream becomes trapped among growing plants. Repeated floods can build those deposits layer by layer. Over long periods, this process helps create broad muddy floodplains that remain in place long enough for more vegetation to become established. Scientists can see the consequences in both modern rivers and ancient rocks. Studies comparing vegetated and unvegetated meandering rivers have found that channels without plants can migrate sideways much faster. After differences in river size are taken into account, unvegetated meanders can shift at rates approaching roughly 10 times those of comparable vegetated channels. A bend in a river moves because flowing water usually erodes the outside of the curve, while sediment accumulates along the inside. Where roots strengthen the outer bank, that movement slows. The bend can remain recognizable for much longer. Vegetation therefore stabilizes meandering rivers, although it is not required to create them. Curved channels can develop through the physics of flowing water and sediment alone. What plants change is the speed at which those bends wander across the landscape and the amount of fine sediment that can remain stored around them. The Paleozoic rock record shows this transformation unfolding as land plants became larger. Before widespread vegetation, broad sand-rich channel systems were common. During the Silurian and Devonian, increasingly rooted landscapes became associated with thicker muddy floodplains and a wider range of river forms. Channels could become more confined because their banks resisted erosion. Floodplain surfaces persisted longer between major disturbances. By the time tree-sized vegetation appeared, rivers were interacting with biological structures far larger than the small plants of the Ordovician. Wood introduced an additional force. A large fallen stem could become lodged across part of a channel. Branches caught against it could collect more debris. Sediment then accumulated around the obstruction, while flowing water was redirected toward another part of the river. Modern rivers still create these log jams, and ancient forests would eventually make woody debris a regular part of terrestrial waterways. That change created physical habitat as well. Pools could deepen beside obstructions. Quiet patches of water formed behind debris. Sediment accumulated in new places, allowing plants to establish themselves on surfaces that might previously have remained unstable. By the Middle Devonian, the spread of increasingly large vegetation was therefore affecting landscapes at a scale far beyond individual stems or roots. A plant could draw water from the ground through vascular tissue, while its roots helped determine how nearby water moved across the land. This combination gave plants a much firmer foundation for increasing their size. The remaining opportunity was above them. Sunlight reached terrestrial plants mainly from overhead, and every additional centimeter of height offered a chance to intercept that energy before neighboring vegetation could use it. As rooted ground became more stable, evolution increasingly rewarded plants capable of building upward. Once plants could remain anchored in the ground and move water through internal tissues, growing upward offered a powerful advantage. Sunlight reaches land mainly from above, so even a small increase in height can place one plant in brighter conditions than its neighbors. A taller stem can intercept light before that light reaches vegetation below. Over many generations, this created steady evolutionary pressure toward greater height in places where plants were competing closely together. The advantage was especially important as vegetation became denser during the Devonian period. Low plants that had once received direct sunlight could begin spending part of the day in shade. Individuals capable of lifting their photosynthetic tissues higher could produce more sugars through photosynthesis and support additional growth. Height also helped with reproduction. Many Devonian plants still reproduced with spores. Releasing spores several meters above the ground could place them into faster moving air, increasing the distance some of them traveled before settling. Growing taller, therefore, offered advantages both for collecting energy and for spreading offspring. The cost increased with every additional centimeter. Water absorbed near the ground had to travel farther before reaching the highest tissues. This movement through a plant is part of its hydraulic system. Hydraulics simply describes the movement of liquids through tubes and spaces. In vascular plants, most upward water transport occurs through xylem. Water molecules tend to cling to one another, helping form an unbroken column inside the narrow conducting cells. Evaporation from exposed plant surfaces then helps pull that column upward. As stems became taller, keeping this water pathway intact became increasingly difficult. Air bubbles could interrupt the column. Greater distance increased resistance to flow. More photosynthetic tissue also meant a larger surface from which water could escape. The stem itself needed stronger construction. A short, flexible shoot can bend without carrying much weight. A trunk several meters tall must support its own mass while resisting gravity and wind. Plants increasingly relied on strengthened cell walls to meet that challenge. One important material was lignin. Lignin is a complex substance deposited in certain plant cell walls, where it makes them stiffer and more resistant to compression. It became especially useful in the cells that carried water and supported upright stems. The same general innovation helped plants solve two problems at once. Strong conducting tissues could transport water, while also helping the body remain standing. Branching added another layer of complexity. A single vertical stem exposes only a limited area to sunlight. Branches spread photosynthetic surfaces outward, allowing a plant to collect light across a wider space. Early branching systems were simple compared with the crowns of modern trees. Over time, some lineages developed increasingly elaborate networks of stems and branchlets. Photosynthetic surfaces also became larger. Small leaf-like structures appeared in several groups, while other plants evolved broader leaves through different pathways. More surface area could capture more light, although it also increased water loss and placed greater demand on the vascular system. The tree form emerged from these linked pressures. Paleobotanists use the term arborescence for the tree habit, meaning a tall, self-supporting plant body with a persistent main axis. Arborescence evolved independently in several plant groups during the Devonian. That repeated evolution is important. The first trees did not belong to one single lineage that later produced every tree on Earth. Cladoxylopsids developed one solution. Lycopsids produced another. Progymnosperms such as Archaeopteris eventually evolved a body plan with still different anatomy. Each lineage faced the same broad physical problems of light, water transport, support, and reproduction. Natural selection repeatedly produced tall plant forms because the rewards of occupying vertical space were so great. By the Middle Devonian, some of those experiments had crossed a clear threshold. Individual plants were now several meters tall, supported by strong internal tissues and crowned with branching structures. They would have been recognizable as trees from a distance. Up close, their anatomy was far stranger. The earliest tree-sized plants lacked the familiar architecture of an oak, maple, or pine, and some built their trunks in ways that no living tree does today. The first successful tree-sized plants would have looked unusual beside almost any tree alive today. One of the most important early groups was the Cladoxylopsids. Cladoxylopsids were vascular plants that flourished during the Devonian period and later disappeared completely. Their internal anatomy was unlike the solid woody trunk of an oak, pine, or maple. One famous example comes from Gilboa, New York. Fossil tree bases had been found there since the 1870s. Paleontologists called them Eospermatopteri. But for more than a century they did not know what the complete plant looked like. The preserved bases showed that something large had once grown there. Its upper body remained a mystery because trunks, crowns, and underground parts are rarely fossilized together. A major clue appeared when researchers studied exceptionally complete fossils from nearby Schoharia County. These specimens connected an Eospermatopteris-type trunk with branches belonging to a plant already known as Watieza. The discovery allowed scientists to reconstruct the complete tree. It stood at least roughly 8 meters tall. That is close to the height of a 2-story building and far above most of the low vegetation that had covered terrestrial landscapes earlier in the Devonian. Its crown looked very different from a modern leafy canopy. Large branches extended from the upper part of the trunk and divided repeatedly into smaller structures. They carried the plant's photosynthetic and reproductive tissues. Broad modern leaves were absent. The whole crown probably had a somewhat fern-like appearance, although Watieza was not a true fern. Those large branches also appear to have been temporary. As they aged, the tree shed them from the trunk and replaced them with new growth near the top. A mature stand of these trees could therefore deposit substantial quantities of plant material onto the ground. That litter provided carbon-rich food for fungi, microbes, and small detritus-feeding animals. The base of Watieza was equally unfamiliar. Instead of sending large, repeatedly branching woody roots deep into the ground, the expanded bottom of the trunk produced many narrow anchoring roots. At fossil sites related to this tree form, these roots typically spread outward through relatively shallow sediment. They were effective enough to hold an 8-meter plant upright, but they explored far less soil than the extensive root networks developed by later trees. Cladoxylopsids also built their trunks in an unusual way. Rather than producing one continuous cylinder of wood like many modern trees, some members of the group contain numerous separate strands of water-conducting tissue arranged within softer material. Other Cladoxylopsid fossils show that these strands could grow in complex patterns as the trunk widened. Evolution had reached a large, upright tree form, using construction methods that have no close equivalent in living forests. This helps explain why paleobotanists use the word tree carefully. A tree is primarily a growth habit. It describes a plant that becomes tall, remains upright, through strong supporting tissues, and maintains a substantial above-ground body. It does not describe one close biological family. During the Devonian, several unrelated groups independently evolved this general architecture. Wattieza and its relatives represented one successful experiment. Later in the Devonian, Archaeopteris developed a different strategy. It produced true wood, more like that of later seed plants, and supported a much more extensive branching root system beneath the ground. Those differences would become important as forests began affecting larger volumes of soil and rock. Before that, later architecture appeared. Cladoxylopsids had already crossed a major ecological threshold. Plants several meters tall could now stand above smaller vegetation, cast shade, and continuously drop organic material around their bases. When enough of these trees grew close together, the result became something far more influential than a collection of unusually tall plants. Entire patches of the Devonian landscape began functioning as forests by the Middle Devonian. Some tree-sized plants were growing close enough together to change entire patches of land. One of the clearest examples comes from the Hangman Sandstone Formation along the coasts of Devon and Somerset in southwest England. The rocks there date to the Eifelian stage of the Devonian period, between roughly 393.3 and 387.7 million years ago. In 2024, researchers describe fossil evidence from these rocks as the oldest known example, preserving the positions of standing trees across a forest landscape. That makes it several million years older than the famous fossil forests of New York. The dominant tree was Calamophyton. Calamophyton belonged to the Cladoxylopsids, the same broad extinct group that produced other strange early trees during the Devonian. Reconstructions suggest these plants commonly stood around 2 to 4 meters tall. They were therefore much shorter than many mature trees today, yet they still rose far above most earlier land vegetation. Their trunks had an unusual construction. The center was largely hollow. Around that hollow interior ran a ring of stronger supporting and water-conducting strands. This arrangement gave the plant enough strength to stand upright without producing a solid cylinder of wood, like the trunk of a modern oak. The upper branches also looked unfamiliar. Calamophyton did not carry broad leaves. Its branches were covered with many narrow, twig-like structures that performed much of the photosynthesis. A full-grown plant may have looked somewhat like a small palm or tree fern from a distance, although it was unrelated to either group. The forest itself appears to have been surprisingly dense. Researchers found tree bases preserved close together, allowing them to estimate how individual plants had been spaced while alive. That detail matters because a fossil forest requires more than a collection of loose trunks. The positions of the trees have to preserve something about the original community. At Hangman Sandstone, rooting structures and fallen plant remains occur within the same sedimentary system. The trees also produce large amounts of litter. As Calamophyton grew, lower branches were shed and dropped around the trunk. Dead stems and smaller fragments accumulated across the ground. For the first time on this scale, large quantities of plant material were becoming a regular part of the terrestrial surface. That debris could slow flowing water and trap sediment. It also supplied food for fungi, microbes, and small invertebrates living among the plants. The forest occupied a river system that experienced seasonal disturbances. Water repeatedly moved sediment through channels and across nearby surfaces. During floods, trees and their rooting structures interacted directly with that movement. Some sediment piled against vegetation. Plant debris collected in certain areas. Rooted ground could remain intact where nearby loose sediment was disturbed. Geologists call features produced by these interactions vegetation-induced sedimentary structures. They are physical traces showing that living plants altered the way sediment was deposited. The importance of the forest therefore extends beyond the age of the fossils. These trees were already functioning as landscape engineers. Their roots strengthened parts of the ground. Their fallen tissues introduced large pieces of biological material into rivers and floodplains. Dense stands changed how water and sediment moved during disturbance events. The relationship worked in both directions. Floods could damage or bury plants, while the plants changed what those floods were able to do. Around 390 million years ago, forests had become large enough for geology to begin recording their presence directly. The land now contained areas where trees stood close together, produced continuous litter, and altered river sediments around them. Yet Calamophytans still relied on relatively modest underground systems. A more powerful form of tree architecture was developing during the same broad interval. Those later trees would combine large bodies above ground with woody, branching roots that reached much farther beneath the surface, bringing early forests another step closer to the way forests function today. The early forests of southwest England were followed by other communities, built from several different kinds of tree-sized plants. One of the most famous was preserved near Gilboa, a small town in New York State. Fossils from Gilboa first attracted attention during the 19th century. Large sandstone casts of tree bases were found there in the 1870s, still standing in the positions where the plants had once grown. More specimens appeared during construction of the Gilboa Dam and Reservoir in the 1920s. For decades, these unusual bases were among the strongest evidence scientists had for the earliest forests on Earth. They were named Archaeopteris. The bases widened near the ground and ended in many small roots. Their trunks had clearly supported plants much larger than the low vegetation of earlier Devonian landscapes. The missing upper portions made their complete appearance difficult to reconstruct. That changed when better fossils were found elsewhere in Schoharia County. Some preserved a tall trunk connected to a crown already known from fossils called Watieza. The connection showed that Watieza and the mysterious Archaeopteris bases belonged to the same general tree form. Associated fossils indicate that these cladoxylopsid trees could stand at least 8 meters tall. Their crowns were made from large branching structures that were eventually shed as complete units. Those fallen branches would have accumulated around the trees as litter, adding large pieces of organic material to the forest floor. Gilboa became even more informative when part of the original quarry surface was uncovered again in 2010. Researchers mapped about 1,200 square meters of the ancient forest floor. The exposed surface preserved numerous Archaeopteris root systems, where the trees had actually grown. The map showed a forest more complicated than a simple stand of identical primitive trees. Between the Cladoxylopsids were long woody structures belonging to another group, called Aneurophytalian progymnosperms. Progymnosperms were extinct vascular plants that shared important features with the lineage that later produced seed plants. Some of the Gilboa forms grew as large woody rhizomes. A rhizome is a stem that grows horizontally along or beneath the ground. Their shoots may have climbed among the larger trees, using the existing forest structure as support. Researchers also found evidence of tree-sized Lycopsids. Lycopsids are an ancient vascular plant group whose living members include small club mosses. Some extinct members eventually became enormous trees, especially later during the Carboniferous period. Gilboa therefore contained at least 3 very different plant growth forms sharing the same forest. The dominant Eospermatopteris trees rose several meters overhead. Woody progymnosperms occupied the spaces around and perhaps among them. Tree-sized Lycopsids added another kind of upright plant architecture. Below this vegetation, the rooting layer remained relatively shallow. Eospermatopteris relied on many small roots extending from its broad base rather than the large woody root systems familiar beneath modern trees. The ancient soil at Gilboa shows limited deep root penetration. This fits a wet coastal plain environment where the forest was periodically disturbed. Individual trees could fall or die, while new plants established themselves in the openings. Every shed what is a branch changed conditions near the ground. Dead plant material supplied food for decomposers and detritus-feeding arthropods. Branches created small sheltered spaces as they accumulated. Different plant heights also began dividing the environment vertically, with conditions near the forest floor differing from those around the upper crowns. A forest had become more than closely spaced trunks. Its architecture was producing several habitats within the same patch of land. Gilboa remained one of the most important windows into this transition, even after older forest evidence was identified in southwest England. It shows how quickly forest communities became diverse once plants learned to build tree-sized bodies. Yet the dominant Gilboa trees still explored only a limited depth beneath the ground. A younger Devonian forest preserved elsewhere in New York contained trees with woody roots spreading meters from their trunks and reaching far deeper into ancient soil. That underground expansion would give forests a much stronger hold on the continents. A different kind of forest was developing in New York only a few million years after the earliest known stands of Cladoxylopsid trees. Near the town of Cairo, paleobotanists uncovered an ancient forest floor dating to about 386 million years ago. The exposed surface covered more than 3,000 square meters, large enough for researchers to map the positions of individual root systems across part of the original landscape. Several kinds of trees had once grown there. Some belonged to Cladoxylopsids, preserving the older tree design already known from places such as Gilboa. Others were associated with Archaeopteris, a plant whose anatomy brought Devonian forests much closer to the structure of later wooded ecosystems. Archaeopteris was a progymnosperm. That term describes an extinct group of plants that produce true wood similar in important ways to seed plants, while still reproducing with spores. Seeds had not yet become the main reproductive strategy of forests. Archaeopteris released spores instead, even though much of its body already looked more advanced than the Cladoxylopsids growing beside it. Its trunk was woody. Branches spread outward from a persistent main stem. Flattened photosynthetic structures covered those branches, giving the crown a much larger surface for collecting sunlight than the narrow twig-like structures of many earlier trees. The most important difference at Cairo was hidden underground. Some Archaeopteris root systems extended as much as roughly 11 meters across. That is wider than many rooms and small houses. The roots repeatedly divided into smaller branches as they spread away from the trunk. They also penetrated much deeper than the shallow anchoring systems common among Cladoxylopsids. In mapped examples, root traces reach roughly 1.2 to 1.6 meters down into the ancient soil. For a Devonian forest, that depth represented a major change. A tree, with roots extending several meters outward, could draw water from a much larger volume of ground. It could reach mineral nutrients beyond the narrow zone immediately surrounding the trunk. During dry periods, deeper roots also had access to moisture that remained below the surface after the upper soil had begun to lose water. The woody construction of those roots made them persistent. They could support large above-ground bodies for years while strengthening the soil around them. Repeated branching also increased the total amount of contact between the plant and the underground environment. A larger root system demanded more energy to build. Photosynthesis in the crown had to produce enough sugars to maintain the trunk, branches, and extensive tissues below ground. That investment paid off by giving the tree access to more water and nutrients, which could then support further growth above the surface. Archaeopteris therefore linked a large crown with a large underground feeding network. Its roots also changed the ground around them. They entered deeper layers of sediment, introduced organic carbon below the surface, and created pathways for water and microorganisms. When portions of the root system died, their remains became part of the developing soil. Minerals that had once been beyond the reach of shallow plants were now exposed to biological activity more than a meter underground. Cairo shows that this more advanced strategy did not appear after every older tree design had vanished. Cladoxylopsids and Archaeopteris existed within the same broad forest community. Several approaches to being a tree were overlapping in time. Some plants relied on relatively shallow rooting and unusual trunk construction. Archaeopteris invested heavily in woody tissues and extensive branching roots. That mixture makes the Middle Devonian feel less like a straight march toward modern forests and more like a period of rapid experimentation. By about 386 million years ago, trees were occupying a much larger part of the environment than their trunks alone would suggest. Their crowns spread through the air. Woody roots reached meters across the ground and more than a meter beneath it. Between those two spaces, forests began creating layers of shade, moisture, litter, shelter, and food that had never existed together on this scale. The terrestrial world was becoming three-dimensional in a completely new way. As early forests became taller and denser, the space between their roots and crowns began filling with new kinds of habitat. Even a partial canopy changed conditions near the ground. Leaves and photosynthetic branches intercepted sunlight before it reached the surface, creating patches of shade that stayed cooler during the day. Reduced sunlight also slowed evaporation. Moisture could remain longer around roots, fallen branches, and low vegetation than it would on an exposed floodplain. That mattered greatly to small animals with limited protection against drying. By the Middle and Late Devonian, arthropods had already been living on land for tens of millions of years. Their surroundings were now becoming far more complex than the low vegetation occupied by early forms such as Archaeopteris. A forest offered sheltered surfaces along trunks and branches. The ground beneath it contained plant litter, fungal growth, and decaying woody material. Roots created cracks and pockets beneath the surface. Each layer had different conditions of light, humidity, temperature, and available food. The continuous supply of litter was especially important. Early cladoxylopsid trees regularly shed large branch systems. Archaeopteris produced woody branches and other tissues that could also accumulate after falling. Once this material reached the ground, its stored carbon became available to organisms that could break it apart. Detritivaries fed on dead plant matter and helped fragment it into smaller pieces. Fungi extended hyphae through the remains and released enzymes that broke large biological molecules into simpler compounds. Bacteria and other microbes continued the process at still smaller scales. Decomposition returned nutrients to the developing soil, where living roots could eventually absorb some of them again. This created a more complete terrestrial nutrient cycle. Carbon fixed from the atmosphere entered plant tissue through photosynthesis. Some of that tissue later fell to the ground and became food for decomposers. The released nutrients could support new growth in the same forest. Arthropods expanded into these new settings. Millipede-like animals could move through damp litter and feed on decaying material. Early arachnids, including extinct spider-like predators called trigonotarbids, were present during the Devonian. They had jointed legs and bodies divided into sections, and some probably hunted smaller arthropods among vegetation and ground debris. Mites occupied still smaller spaces. Primitive hexapods, distant relatives within the broader line that later produced insects, were also part of Devonian terrestrial communities. A forest therefore created several ecological levels at once. The subsurface contained roots, fungal networks, and microbes. The soil surface collected litter and supported detritivores. Higher stems and branches created living surfaces above the ground, while shaded spaces between larger plants allowed smaller vegetation to persist under different light conditions. Fallen wood added another form of structure. A large branch lying across the ground could trap sediment during a flood. Water might collect beside it. Small animals could shelter beneath it, while fungi and microbes slowly entered the dead tissues. As more woody material accumulated, the forest floor became physically more complicated. Rooted vegetation also stabilized wet areas along river margins. Sediment trapped between stems could build slightly raised surfaces. Other depressions remained damp or periodically flooded. The result was a patchwork of microhabitats separated by only a few meters. The animals living there could specialize on different foods or moisture conditions, rather than all occupying the same exposed surface. This was a major ecological consequence of tree-size growth. Forests increased the amount of usable habitat without requiring the continents themselves to become larger. A single patch of land could now contain biological activity beneath the soil, across the ground, and several meters into the air. The tiny arthropods that had first entered terrestrial environments more than 400 million years ago were now living inside ecosystems with shade, litter, woody structure, and persistent root zones. And as these forests spread across larger areas, their influence could no longer remain confined to the organisms living among them. Millions of roots were reacting with minerals beneath the ground, while vast amounts of photosynthetic tissue were drawing carbon dioxide from the atmosphere. The forest was beginning to operate as part of the chemistry of the planet itself. Once forests spread across larger parts of the Devonian continents, their effects reached far beyond the shaded ground beneath their branches. Every green surface was carrying out photosynthesis. During that process, plants absorbed carbon dioxide from the atmosphere and used sunlight to build carbon-rich sugars. Some of that carbon became wood, roots, spores, and other plant tissues. When those tissues died, most of the carbon eventually returned to the atmosphere as organisms decomposed them. A small portion followed a different path. Plant material could become buried in waterlogged sediment or carried into places where decomposition slowed. If organic carbon remained buried for long periods, the carbon dioxide used to build that tissue stayed out of the atmosphere. Over millions of years, repeated burial could influence the balance of carbon between the atmosphere, rocks, oceans, and living organisms. This process is also connected to oxygen. Photosynthesis releases oxygen, while carbon is fixed into organic matter. When that organic matter is later decomposed, much of the oxygen is effectively used again. Long-term burial allows some of that oxygen to remain in the atmosphere. Forests therefore contributed to changes in atmospheric oxygen, although they did not create oxygen on Earth. Large amounts of oxygen had already accumulated long before the first trees appeared. Roots added another powerful effect below the surface. Many rocks contain silicate minerals, a broad group built around silicon and oxygen. When these minerals react with water and carbon dioxide, they slowly break down through a process called silicate weathering. Root respiration increases carbon dioxide in soil water. Fungi and microbes around the roots release additional compounds that can speed some mineral reactions. As Devonian rooting systems became deeper and more widespread, a much larger volume of rock and sediment came into contact with this biological activity. Scientists have linked the expansion of land plants with major changes in chemical weathering. The exact size of that effect remains difficult to measure, because weathering also depends on temperature, rainfall, rock type, and tectonic activity. Still, forests clearly added a new biological force to the process. Weathering can affect climate because some reactions involving silicate minerals ultimately remove carbon dioxide from the atmosphere over long timescales. Lower carbon dioxide reduces part of the greenhouse effect. During the Devonian, atmospheric carbon dioxide declined substantially from earlier Paleozoic levels, while climate also experienced important cooling phases. Forests likely contributed to that broader trend alongside changes in mountain building, ocean circulation, volcanic activity, and the arrangement of the continents. The growing vegetation also changed the movement of water. Trees absorb water through their roots and release much of it through tiny openings in their leaves or other photosynthetic surfaces. That release is called transpiration. In a large forest, enormous quantities of water can move from soil into the atmosphere this way. Some later returns as rain. Forests therefore become part of the water cycle as well as the carbon cycle. Their roots also influence how long sediment stays on land. A vegetated floodplain can hold mud and sand for longer periods because roots strengthen the ground and plant stems slow floodwater. Longer sediment residence gives minerals more time to weather before they are carried away. Organic matter can also remain trapped within those sediments. By the Middle and Late Devonian, forests were tying together several parts of the Earth system at once. Photosynthesis changed atmospheric carbon. Roots altered mineral reactions. Vegetated floodplains changed the storage of water and sediment. The combined result was a planet where living organisms were helping control processes once governed mainly by geology and climate. Those effects did not stop at the edge of the continents. Rivers collected dissolved minerals and fine particles from rooted landscapes and carried them toward lakes, estuaries, and oceans. As forests changed what entered the rivers, they also began changing what eventually reached the sea. As rooted plants changed the chemistry of the continents, rivers carried some of those changes toward the oceans. One of the most important elements involved was phosphorus. Phosphorus is essential for living cells. It is used in genetic material, cell membranes, and the chemical system that transfers energy inside cells. In the oceans, phosphorus can also act as a limiting nutrient. That means biological growth may be restricted simply because there is not enough available phosphorus to support more organisms. Roots and the microbes living around them can help release phosphorus from minerals during weathering. As Devonian rooting systems became deeper and more widespread, more rock was exposed to this biologically active zone. Some of the released phosphorus entered streams and rivers. From there, it could eventually reach coastal waters and the open sea. That process became especially important during the late Devonian. Around 372 million years ago, Earth experienced a major environmental crisis known as the Kellwasser event. It was part of a longer interval of biological disruption near the end of the Devonian period. Marine ecosystems were hit especially hard. Many reefs collapsed, and numerous groups of ocean animals declined sharply. One of the clearest signs of stress comes from layers of dark rock known as black shale. Black shale commonly forms where large amounts of organic material settle into water with very little oxygen. Geologists have found these dark deposits in many regions of the world from the same general interval. They point to widespread marine anoxia. Anoxia means that dissolved oxygen in the water falls to extremely low levels or disappears entirely. The connection to land plants begins with nutrients. If rivers carry more phosphorus into the ocean, microscopic photosynthetic organisms can grow more rapidly. Greater biological production near the surface creates more organic material. When those organisms die, part of that material sinks into deeper water. Bacteria then break it down. Decomposition requires oxygen. If enough organic matter reaches deeper water, bacteria can consume oxygen faster than currents, and surface mixing are able to replace it. Large areas of the ocean can then become poorly oxygenated. Animals living on the seafloor are especially vulnerable because they cannot simply move into the atmosphere for oxygen. Fish and other mobile animals may escape some affected areas, but widespread or repeated anoxia can remove enormous amounts of usable habitat. Computer modeling published in 2023 tested whether the spread of land plants could have contributed to this process. The models showed that stronger weathering and increased phosphorus transport from vegetated continents could plausibly help produce widespread oxygen loss in Devonian oceans. That result gives the forest hypothesis a clear physical mechanism. It does not make forests the single cause of the Kellwasser crisis. The Devonian Earth system was changing in several major ways at the same time. Large volcanic episodes may have released gases and nutrients that affected both climate and ocean chemistry. Changes in sea level altered the amount of shallow marine habitat, Ocean circulation could influence where oxygen-rich surface water reached deeper regions. Recent geological studies continue to find strong evidence that anoxia played an important role during these extinction intervals, while the exact combination of causes remains debated. The safest picture is therefore one of interacting pressures. Forest expansion changed weathering on land. Rivers transferred some of those products toward the sea. Volcanism and climate shifts could amplify or modify the same ocean systems. The result was a series of environmental stresses operating across enormous areas. The scale of the transformation is striking. Roughly 100 million years earlier, land plants had been small enough to remain close to damp ground. Their rooting structures were shallow. By the late Devonian, forests were changing erosion, nutrient transport, and the chemistry of water reaching the oceans. A biological innovation that began on exposed terrestrial surfaces had become connected to marine environments thousands of kilometers away. That connection between land and sea still exists today. Nutrients released from soil continue to travel through rivers. Excess phosphorus can still trigger blooms and oxygen-poor water in modern lakes and coastal regions. The mechanisms are ancient, even when the landscapes around them have changed completely. By this point in Earth history, trees had become part of a planetary system linking soil, rivers, atmosphere, and ocean. The world that existed before them had been transformed so deeply that its traces now survive mainly in fossils, ancient soils, river deposits, and chemical signals locked inside rock. The Devonian world eventually became so different from the one Prototaxites had known that the earlier landscape can be difficult to reconstruct from modern experience. When Prototaxites Tahiti lived at Rhynie around 407 million years ago, most terrestrial vegetation remained low. Small vascular plants rose only centimeters above the ground. Arthropods moved among damp patches of vegetation and decaying material. Soils were developing, yet deep woody root systems had not spread through them. Riverbanks lacked the dense reinforcement that forests would later provide. Across some environments, sediment could shift far more freely during floods. The 2026 study of Prototaxites therefore belongs to a much larger story. Its unusual chemistry and tube-filled anatomy suggests that this organism may have belonged to an extinct lineage of complex life, with no close living equivalent. Its strangeness fits the time in which it lived. Terrestrial ecosystems were still assembling combinations of organisms and body plans that would later disappear. Over the following tens of millions of years, plants changed both above and below the surface. Vascular tissue allowed water to travel farther through upright stems. Roots expanded into deeper layers of sediment and released compounds that helped alter minerals. Tree-sized plants began standing several meters high. By roughly 390 million years ago, dense stands of Calamophyton were already forming recognizable forests in what is now southwest England. Later forests included trees such as Archaeopteris, whose woody roots could spread many meters from the trunk and penetrate more than a meter into ancient soils. Those developments changed the behavior of entire landscapes. Rooted riverbanks resisted erosion more effectively. Floodplains retained larger amounts of fine sediment. Plant litter accumulated in quantities that earlier terrestrial ecosystems had never produced. Shade created cooler and moister places close to the forest floor. Branches and trunks added physical habitat above the ground. while roots expanded the living environment beneath it. Modern rivers still show the mechanical effects of vegetation. Where banks are densely rooted, soil usually resists flowing water more strongly. When vegetation disappears, exposed sediment can become easier to erode and channels may shift more rapidly. The comparison is imperfect because today's rivers contain hundreds of millions of years of later biological and geological history. Even so, the same basic physics helps explain why the spread of roots during the Paleozoic left such a clear mark in ancient river deposits. Forests also became part of larger chemical cycles. Photosynthesis moved carbon from atmospheric carbon dioxide into plant tissues. Roots and their microbial partners increase contact between life and mineral surfaces. Rivers carried dissolved nutrients from those landscapes toward the sea. By the late Devonian, these processes may even have contributed to episodes of marine oxygen loss, when additional phosphorus increased biological productivity in the oceans. The journey from small land plants to forests therefore changed much more than the appearance of the continents. Many features that now seem ordinary depend partly on biological systems built over immense spans of time. Agriculture relies on soils whose structure and fertility are maintained by roots, microorganisms, organic matter, and mineral weathering. Settlements are commonly built beside river systems, whose banks and floodplains are influenced by vegetation. Human infrastructure rests on terrestrial surfaces that have spent hundreds of millions of years interacting with life. That history makes the phrase "earth before trees" much larger than the absence of trunks and branches. The continents were still becoming more deeply rooted. Their soils were gaining complexity. Rivers were adjusting to stronger banks and larger amounts of organic debris. Terrestrial habitats were expanding upward into crowns and downward through branching roots. The strange columns of Prototaxites belonged near the beginning of that transformation. Some members of the genus reached roughly 8 meters tall, while ordinary land plants around them remained far smaller. For a time, one of the tallest organisms on land may have belonged to a lineage that eventually vanished completely. That helps explain why Prototaxites has been so difficult to classify. Scientists are trying to understand an organism from a terrestrial world that no longer exists, using biological categories shaped mostly by the survivors. Trees eventually filled much of that open vertical space and changed the ground beneath it. The forests around us today are part of the result. And beneath every familiar trunk lies a much older story, stretching back to a time when the continents themselves were still learning what it meant to be covered by life.